Today's first talk is from Dr. Tomi Pastinen, Director of the Center for Pediatric Genomic Medicine at Children's Mercy Hospital, and also affiliated with McGill University. The title of the talk is Interacting Differentially Methylated Regions to Infer Chromatin Looping.
All right. Good morning, everyone. I hope you're hearing me well. So I'll describe our work in the last several months in the 40 Nucleome TCPA grant that was awarded to us last year as the Canadian component of the TCPA program, and I'll describe really the motivation initially. So we have been mapping epigenomes in context of human tissues and diseases, both for the reference epigenome mapping effort within the International Human Epigenome Consortium, but then in more disease gene-oriented programs as well as carrying out epigenome-wide association studies for human disease. And in a lot of these studies, what we and others have been finding is that there seems to be abundance of non-coding regulatory variation that is distal from genes, and recent studies have been starting to look at chromatin conformation as a tool to link these distal regulatory elements to gene targets in disease context. So we have been interested in chromatin conformation assays for a while, but we couldn't really envision how we would be able to scale the assays to directly infer chromatin looping in large numbers of patient samples, for example, given the requirement of relatively high amount of material as well as the high quality of the material. So for epigenome, what we've done and continue to use to infer regulatory differences between samples is use high density methylation analysis across cells and tissues in disease cases and normal controls, or across different cellular conditions such as differentiation or stimulation. And when we compose these large data sets, we have started to observe distal correlations or coordinated changes in methylation, and we wanted to explore in the 4D and TCPA program whether this could be used as a tool to infer methylation, to infer chromatin conformation in larger scale across human populations and disease studies. The attractiveness of methylation as an inference tool comes partly from its versatility as an assay. It's a chemical assay rather than enzymatic or immunologic assay, so we have very low background, and also the requirements of the sample quality and quantity are much reduced compared to most chromatin-based assays. So in the past, what we've done, in addition to methylation variation assessment, we've also done large-scale assessment of chromatin variation across human cells and tissues, and this is from a study that we did 2 years ago, or published 2 years ago with Wellcome Trust Sanger Institute with Nicole Saranso's group, where we actually mapped chromatin variation across post-translational modification of histones in over 600 human samples from 3 different cell populations and 200 donors. We also did methylation variation assessment and expression variation assessment in these same cell types, so to generate a high density data set of correlated chromatin expression and methylation variation. And what we observe for most genes is shown here. We observed that proximal regulatory elements such as promoters show variation that is coordinated across long regions in the cis-regulatory space, and this coordination is, as shown here, as allelic coordination of different regulatory elements in the vicinity of the gene is often spanning much longer regions than shown here. And in this particular study, we did not do chromatin conformation, but we did think that this strong allelic coordination across several elements in the cis-regulatory space would probably represent some sort of physical interactions in many cases. So we've since gone forward and deepened this data set using a high-density methylation sequencing approach, where we've actually targeted all regulatory elements present in circulating immune cells. And these regulatory elements come from several different data sets by roadmap epigenomics and our own data sets on DNase hypersensitivity, as well as other regulatory features. In total, we assay about 5 million CPG sites, and this captures regulatory variation efficiently, and we've applied this now in thousands of samples. And what we, in the first study that we published, what we observed that about 20% of these methylation changes at regulatory elements show allelic behavior, so the parental copies of the genome behave differently. This provides large resource of looking at allelic interactions across the genome. And we also have been able to show that if you map histone variation and compare that to methylation variation, most of the histone mark variation in population is captured by methylation variation, but in addition, methylation variation captures several-fold more population variation in, for example, circulating immune cells. So we believe that the method is efficient in comprehensively capturing variation across samples. But the question is, can we use that in inferring physical interactions in the genome? So we used data from a separate study published for human immune cells on promoter capture high-C, and looked at whether the CPG correlations in independently verified loops showed more correlation than background. The background is shown in red here, and the loops, the correlations from the loops are shown in white bars, and we see many more correlations as compared to matched background within loops. So there seems to be abundant genome-wide evidence that distal methylation correlations actually, in many cases, could represent physical interactions. However, most of the distal methylation correlations that we observe in these large data sets don't have independently verified loops, and similarly, many loops don't have independently confirmed methylation in our data. So at present, or using this approach of our previous mapping data and external data, we are not able to derive methods to detect false positives and negatives, or algorithms to predict which methylation, coordinated methylation events are actually representing interactions. I would note that in the same data set, we also correlated distal allelic behavior of methylation and also see enrichment as compared to matched background of significant correlation in allelic behavior in previously shown loops. So both allelic and total methylation can be used to infer interactions. So in the 4DN program, what we aim to do now is rather than using these literature-based comparisons to previously published loops to generate high-density, high-C data in parallel with methylation data. And we started, since our labs were naive to high-C before, so we started initially with the endothelial cell model, which can be induced using TNF-alpha. So this to start optimizing the high-C, and we used the Decker lab protocol, and we were able to generate relatively high quality high-C libraries based on the HICcup QC pipeline. And this initial data set we generate in-house to look at looping versus methylation. We had both TNF-alpha induced cells and control cells. And I guess as expected, the TNF-alpha induction doesn't really switch chromatin compartments. However, we do see evidence of local looping changes upon TNF induction, but overall the changes are relatively small. So this is not an optimal system to study how well variation in looping correlates with methylation, but we are able to show that long distance looping detected in the endothelial cell system, as shown here is an example of a 1.7 megabase loop across, these are ATAC-seq peaks across two regulatory elements, shows high methylation correlation, and another example of a slightly shorter interaction loop, again showing high methylation correlation. So at least we are able to replicate in the same samples some of the external data that I showed previously for the CD4 plus T cells. Overall, however, the enrichment of positive loops is relatively minor, so it's represented by this small hump here, while it's the enrichment of significant positive correlations is significant. It's not sufficient to start to independently verify and develop models in this system. So we need more variation within the sample or between samples to verify that the high C and methylation variation is concordant. And what we've started to now use is an adipose tissue model where we have independently collected a lot of epigenome and transcriptome variation data from primary tissues, and then we have a cellular differentiation model where we also have population variation component from 30 different donors, where we can look at high C, which is not possible in these tissue samples. And similar to the other data that I showed you, if we look at just correlation of distal methylation with gene expression, if the correlation with promoter and putative enhancer methylation is high, we observe significant enrichment of methylation versus expression correlation, suggesting that this is a true interaction. And to validate that, we've now started to develop high C looping data from the adipose derived adipose cells, and we're using the dovetail low input high C protocol to achieve that, and this has recently been successful. So now we're able to ask directly questions of whether the methylation variation we observe in adipose tissues is overrepresented, or the coordinated methylation variation across distal sites is overrepresented in high C loops, and we see significant evidence of enrichment again of these coordinated CPG events with CPG correlations with independently verified loops in the same cell system. So this is promising, and we have been able to validate several distal CPG correlations now using the first Hi-C data that we've generated in the adipose cells. Here's an example of the one key regulator in adipose tissue, SHIF1, and distal loops that were initially discovered as distal methylation correlations and then verified by Hi-C in the derived adipocytes. Another example from the KLF9 locus showing similar results. So overall, we only have a few individuals now with Hi-C data in the adipose tissue, and we are expanding that to be able to have more variation between individuals and within individuals through differentiation in this system. But even now we can already start to ask questions about how do coordinated methylation events that are within loops behave. And what we see is that in enhancers that are low methylated regions, the highest correlated CPG sites are close to the midpoint of the enhancer, whereas in promoters that are relatively unmethylated, the highest correlations are seen at the edges of the promoter regions, and this is because the midpoints of promoters are relatively non-dynamic, close to zero methylation. So this indicates that in order to detect dynamic variation between loops in promoters and enhancers, we need higher depth whole genome bisulfite sequencing. So within the 4DN program, now that we have several Hi-C protocols working in our cell models, we are pursuing in the next year, and which is ongoing now, developing deep Hi-C data and deep parallel whole genome bisulfite sequencing data in the same cells, which will allow us to build better models for understanding which type of interactions or coordinated methylation events are representing true physical interactions in genome-wide manner. What we initially wanted to do, and I didn't show you the negative data, is our initial plan was to generate high-resolution Hi-C maps using enrichment techniques such as promoter capture or high-chip approach, but we haven't been really able to get more than 2- to 4-fold enrichment of targeted sites. So we've given up on that and are generating shotgun Hi-C data in slightly smaller number of samples that we initially targeted. And we've decided that initially we thought that we would use several different differentiation protocols in the mesenchymal stem cells, from mesenchymal stem cells to adipocytes, chondrocytes, or osteoblasts, but now we'll focus on the adipocytes and generate higher density data for both on Hi-C and by whole genome bisulfite sequencing the remainder of the project. So overall, this is what's planned for the coming year, and I would note that the 4D Nucleome project is related to the standard epigenome mapping program that we apply in the same cell system. So in addition to the deep whole genome bisulfite sequencing, we have chromatin marks and ATAC-seq as well as RNA-seq data collected, and we can correlate that to the deep Hi-C data that we'll generate in this system to develop models on interactions and validate interactions using Hi-C. And obviously we want to disseminate this data through the 4D and Nucleome for others to explore how coordinated epigenome variation is reflected in physical looping. So that's all, and I'm happy to answer questions.
Thank you. That's wonderful. You've given us a lot to digest, as it seems you're going exactly where the 40N consortium was envisioned to go in the future in terms of translating this into looking at people in different tissues and through development. So let's take questions now from anyone.